designing targeted cancer therapies today, uh, it often feels like trying to take out a heavily reinforced bunker with a guided missile that is it's simply too massive to penetrate the outer walls.
Right, because the targeting system might work flawlessly, but if the missile gets stuck at the perimeter, I mean, the target inside just survives.
Exactly. And we still largely rely on static weapons. You deploy them and, just hope the physics play out in your favor. But what if we didn't just fire a static weapon? What if we could actually program drugs the exact same way we program computers?
Well, we'd essentially be transitioning from just, delivering a toxin to what researchers are now calling computed drug delivery.
Computed drug delivery.
Yeah. The drug itself actually evaluates its surroundings, makes a localized decision based on biological parameters, and then, and only then, executes a programmed command.
And that concept is the core mission of our deep dive today.
Wow.
We're breaking down a genuinely mind-bending paper that was published in Nature Biotechnology in March 2026.
It's an incredible piece of research.
It really is.
Mm-hmm.
The researchers figured out how to use DNA logic gates, which function just like the Boolean A&D gates in a microchip to amplify drug toxicity on specific cancer cells. And they do this while completely ignoring healthy bystander cells.
Which is the holy grail of oncology, really. But to appreciate why computing a drug's delivery is such a massive leap, we need to talk about those bunker walls you mentioned.
Right. We have to look at the current gold standard in targeted therapy, which are antibody drug conjugates, or ADCs.
Yeah, if you're following oncology at all, you've likely heard of ADCs. Things like Enhertu, they operate on a really elegant premise.
You have a monoclonal antibody, right, which is this massive protein engineered to seek out a specific biomarker on a cancer cell.
Exactly. And attached to that antibody is a highly potent chemotherapy drug connected by a chemical linker. So the antibody acts as a homing beacon.
It just drags the toxic payload directly to the cancer cell, hopefully sparing the rest of the body.
Right. The homing beacon finds the target and delivers the poison, but they're hitting a pretty severe physical limitation when it comes to solid tumors.
The guided missile is too big.
It is a problem of sheer scale. An intact monoclonal antibody weighs around 150 kilodaltons.
Okay, to put that in perspective for you, a standard small molecule drug like, say, aspirin, is about 0.18 kilodaltons.
So an ADC is nearly 1,000 times larger. And because they are so incredibly massive, they diffuse very, very slowly through the dense tissue of a solid tumor.
It's basically like trying to drive an 18-wheeler truck through the narrow, twisting cobblestone streets of a medieval city.
That's a great way to picture it.
Like, it's carrying exactly what the town center needs, but the truck fundamentally cannot fit through the alleys.
Close, but I'd say it's more like an 18-wheeler covered in industrial superglue.
Oh, wow. Okay.
Yeah, the phenomenon in oncology is known as the binding site barrier. Because these ADCs are so massive and they bind so tightly to their targets, they essentially saturate and get permanently stuck to the very first cancer cells they bump into on the outer edge of the tumor.
Right, so the cells deep inside the tumor mass never even get exposed to the drug.
Exactly. The perimeter absorbs the entire attack.
And to add to the payload problem, an 18-wheeler that size can only safely carry a tiny amount of highly toxic cargo before the structure just destabilizes, right?
Yeah, that's a huge bottleneck. Most traditional ADCs carry maybe 2 to 4 drug molecules. Some of the newer ones push it to 7 or 8, but
We're still talking about single digits.
right, single digits. And that limitation is really the driving force behind this new research. The researchers recognize that if massive antibodies are getting caught at the binding site barrier, the physical solution is to send in much smaller scouts.
So instead of full-size antibodies, they utilize structures called affibodies and aptamers.
It's exactly. Which are essentially affinity proteins and nucleic acids that act just like antibodies, but they're a fraction of the size.
We're talking less than 10 kilodaltons, right, compared to the 150 of an ADC.
Yeah, they're tiny. They slip right through that dense tumor matrix.
But logically, I mean, you can't just slap a highly toxic chemotherapy drug onto a tiny affibody. If they're small enough to bypass the tumor matrix, they're small enough to just wash all over the body, right? Penetrating healthy tissue, too.
That's the danger, yes.
And since they're tiny, they can't carry enough payload to kill the cell anyway. You'd just be flooding the system with microscopic, highly toxic, completely ineffective darts.
That is the exact paradox that they needed to solve. You need the deep tissue penetration of a small molecule, but you also need the high specificity and high payload capacity of a massive ADC.
Which seems impossible.
Until you bring in the biological logic gate. The researchers designed what they call a split initiator using DNA. Let's say we're targeting a cancer cell that expresses two specific biomarkers on its surface.
Okay, for the sake of understanding the mechanism, let's look at EGFR and PD-L1. Those are very common targets.
Perfect. So we take one tiny affibody designed to bind to EGFR, and we attach one half of a specific DNA strand to it.
Just half.
Just half. Then we take a second tiny scout, maybe an aptamer, that binds to PD-L1, and we attach the other half of the DNA strand to that one.
Okay, so both scouts are deployed, and they easily penetrate deep into the tumor because of their small size.
Right. If one scout finds EGFR on a cell, it binds. If the other scout finds PD-L1 on that same cell, it binds.
And when both are bound to the same cell membrane, they're forced to sit in close physical proximity.
Exactly. And that localized concentration allows their respective half strands of DNA to just snap together, forming a complete functional sequence.
Which is known as a three-way junction, or a 3WJ.
You got it.
But wait, let me pause you there. These halves are just floating through the bloodstream. What stops them from just randomly snapping together in the blood or, connecting on the surface of a healthy cell that happens to have just one of those markers?
Well, the researchers had to tune the thermodynamics of that DNA connection perfectly. It's a delicate balance.
How do you tune thermodynamics in DNA?
By experimenting with the length of the DNA stems connecting the halves, they tested lengths of 4, 6, and 8 nucleotides. When the stem was only 4 nucleotides long, the binding energy was just too weak.
So they would bump into each other,
But the repulsive forces of the attached affibodies would pull them apart. The connection failed.
Okay, and if they made it 8 nucleotides long?
The binding energy was too strong. The DNA halves wanted to pair up so aggressively that they would snap together even if only one scout had actually found its target on the cell.
Oh, I see. So the DNA affinity actually overpowered the biological targeting.
Exactly. In computing terms, that causes A&D gate leakage, which is a false positive trigger.
So 8 is too aggressive, firing when it shouldn't. 4 is too weak, breaking apart when it should fire.
Exactly. 6 nucleotides created the perfect thermodynamic balance. At 6 nucleotides, the DNA halves only have enough energy to form the junction if and only if both biomarkers are holding them tightly in place right next to each other on the cell surface.
That's brilliant. If the cell only has EGFR, it doesn't trigger. If it only has PD-L1, it doesn't trigger.
It computes a flawless Boolean function. If biomarker 1 and biomarker 2 are present, then form the junction.
Okay, so the scouts have penetrated the tumor, found the specific dual marker cell, and the A&D gate has snapped together. But we still have a major problem here.
The payload.
Yeah, we just have 2 tiny proteins and a small piece of DNA sitting on a cell. There's no payload yet. How do we amplify this into a lethal attack without reverting back to the massive 18-wheeler truck?
Well, that completed three-way junction is essentially a molecular switch. To flick that switch and deliver the payload, the researchers utilized a mechanism called a hybridization chain reaction, or HCR.
Okay, HCR, how does that work?
Once the A&D gate forms on the target cell, it becomes an active initiator. At this stage, you flood the system with independent DNA structures called hairpins, which are preloaded with toxic chemotherapy drugs.
Wait, so these hairpins are just circulating in the blood, fully armed?
They are heavily armed but completely inert. They're kinetically trapped in a closed loop, meaning they cannot interact with each other or with healthy cells.
Like a loaded spring that hasn't been triggered.
Exactly. But when a hairpin encounters the active initiator on the cancer cell, the initiator unzips the hairpin. And here is the brilliant part of the chain reaction. Unzipping the first hairpin exposes a new sequence of DNA that acts as the key to unzip a second hairpin.
Oh, wow.
Yeah. The second one unzips a third, the third unzips a fourth, and it just cascades continuously.
So instead of a massive drug delivery truck, you've created a system that autonomously manufactures the truck on site.
That's a perfect way to put it.
It's like snapping together a massive scaffold of molecular zip ties. The first one clicks into place on the cancer cell, opening a slot for the next one, building a massive custom polymer structure directly on the target.
And the amplification metrics are staggering. The chain reaction creates a signal amplification of over 100-fold in just 30 minutes.
That fast.
Yeah. In one of their experiments targeting EGFR, they measured a median of 217 propagation steps.
217 steps. Traditional ADCs max out at what, 8 drug molecules?
Exactly.
So this program system is assembling a polymer chain carrying potentially hundreds of drug molecules built layer by layer on the cancer cell itself.
It's a completely different scale of delivery.
But wait, think about the physical structure here. You've just built a massive toxic DNA polymer on the outside of the cell.
Right.
DNA doesn't just phase through lipid membranes. how does the poison actually get inside the bunker to kill the cell?
The cell membrane is indeed a barrier, but cells are dynamic. They constantly sample their environment and recycle their surface receptors through a process called endocytosis.
Where the cell membrane folds inward, right? It basically swallows whatever is attached to the receptors and pinches off to form a little internal bubble.
Exactly. And because the massive DNA chain is physically anchored to the EGFR and PDL1 receptors, when the cell naturally pulls those receptors inside for recycling, it drags the entire toxic polymer in with it.
It's a molecular Trojan horse.
It really is. Once swallowed, that internal bubble, which is called the endosome, merges with a lysosome. And the lysosome is effectively the cell's stomach.
So it's highly acidic and packed with digestive enzymes.
Right. And the researchers attached the toxic chemotherapy drugs to the DNA hairpins using a specific chemical linker. This linker is designed to be cleaved by an enzyme called cathepsin.
Which is waiting in the lysosome.
Exactly.
So the cell swallows the Trojan horse, digests the DNA, and the cathepsin sniffs the chemical linkers, unleashing the payload from the inside out.
And the sheer size of that swallowed polymer creates a kind of time-release mechanism, exposing the internal machinery of the cancer cell to the drug for over 5 hours.
That's incredible.
But the researchers discovered something highly unusual during this phase. They tested different payloads on the hairpins, primarily looking at 2 common ADC chemotherapy drugs, MMAE and DXD.
Okay, 2 different toxins.
Right. And they compared a hairpin loaded with a single copy of MMAE to a hairpin loaded with 3 copies of MMAE.
Well, I mean, basic logic dictates that a hairpin with 3 copies of the drug would be roughly 3 times as toxic as the single copy version.
You'd think so. But the data showed something entirely different. The hairpin loaded with 3 copies of MMAE didn't just deliver 3 times the drug. It was 50 times more active.
50 times.
Yeah, and it drove a 10-fold higher cellular uptake of the massive DNA chain.
Wait, increasing the payload didn't just make it more toxic, it actually changed how the cell physically swallowed the DNA. How does the cargo affect the cell's ingestion mechanism?
It comes down to a biophysical property called log P.
Log P. Okay, what is that?
It measures lipophilicity, or essentially how fat-soluble a molecule is. A high log P means the molecule desperately wants to merge with lipids like fats or oils to escape water. MMAE has a high log P. It's 2.14.
Okay, and what about JAK-STI?
JAK-STI has a low log P of 0.88, making it much more water-soluble.
Right, and the cell membrane is literally a lipid bilayer. It's a wall of fat.
Exactly. So when you load 3 copies of this highly fat-soluble MMAE onto every single step of a 200-step DNA chain, you completely alter the thermodynamics of the polymer.
Oh, I see. You're creating a massive, highly hydrophobic patch on the outside of the cell.
Yes. To escape the watery environment of the body, that massive DNA chain actively partitions itself, literally sinking directly into the lipid membrane of the cell.
It just buries itself into the fat of the cell wall.
Right. And that aggregation forces the membrane to buckle inward, artificially triggering endocytosis. The drug is no longer just passive cargo. The chemical properties of the drug itself become the steering wheel, actively forcing the cell to swallow the entire structure.
That is wild.
And just to prove it, when they tested the exact same 3-copy structure using the water-soluble drug DXD, the enhanced uptake completely disappeared. The process required the specific lipophilic profile of MMAE to drive that aggressive membrane partitioning.
Okay, so we've established an incredibly lethal, self-assembling, self-internalizing weapon. But the critical hurdle in oncology is always collateral damage. A real tumor is a messy mix of malignant tissue and healthy bystander cells.
It is.
So if this chain reaction is so potent, how do we know it won't just spill over and eradicate the healthy tissue sitting right next door?
To test the ultimate precision of the A&D gate in a realistic, messy environment, the researchers set up a mixed culture microscopy experiment. They basically grew 2 types of cells entirely intermingled in a single dish.
Which simulates the tumor environment.
Exactly. For this specific test, they utilized A431 cancer cells, which natively express both EGFR and a second biomarker, PTK7, and they grew these right alongside HeLa cells, which served as the healthy bystanders. The HeLa cells lack that specific dual marker profile.
So they're physically touching each other, sharing the exact same fluid environment.
Right. They applied the logic-gated HCR system loaded with MMAE to the dish and just waited 48 hours.
And what happened?
The microscopy revealed a stark contrast. The target A431 cancer cells were virtually eradicated. The HCR system identified the dual markers, assembled the polymers, and completely destroyed the targets.
And the bystander cells.
The HeLa cells, floating in the exact same chemical bath, proliferated completely normally. They were utterly untouched, zero bystander effect.
Wow. So the AND gate held. It only unleashed the assembly process on the cells that provided the exact 6-nucleotide thermodynamic lock.
It held perfectly. And beyond the precision, the system is fundamentally modular. Because you're working with programmable DNA hairpins, you aren't restricted to a single payload.
What do you mean?
The researchers demonstrated they could actually mix hairpins loaded with MMAE and hairpins loaded with DXD. They were simultaneously assembling combinations of drugs to attack the cell through different mechanisms, which helps prevent resistance.
That is so clever.
And they even proved you don't have to use chemotherapy at all. They loaded the hairpins with chemical tags that capture generic immune antibodies. So basically using the massive DNA polymer to paint a giant target on the cancer cell, recruiting the patient's own immune system to clear the threat.
If you really step back and look at the trajectory here, we are leaving the era of clunky 18-wheeler ADCs that get trapped at the outer edges of tumors. We are entering an era of programmable molecular scouts.
It's a paradigm shift.
They slip deep into the tumor matrix, evaluate the surface of a cell using literal Boolean logic, and if the conditions perfectly match the code, they construct a custom, hyperlethal delivery vehicle on the spot. And then they use the drug's own biophysics to force the cancer cell to eat its own destruction.
Beautifully summarized.
I mean, we aren't just finding drugs anymore.
Right.
We're writing the source code for localized chemical factories.
And, the implications stretch far beyond just this single study. If we can reliably program a high-fidelity ND gate on a cell membrane today using a 6-nucleotide stem, we are quite literally looking at the foundational architecture of molecular software.
Molecular software, it sounds like science fiction.
It does, but the next logical progression is moving beyond simple binary triggers. We will soon be coding complex if-then-else statements into our therapies.
Like evaluating multiple conditions dynamically.
Exactly. Imagine an autonomous drug structure that binds to a cancer cell, evaluates the cell's specific real-time resistance profile, and then dynamically selects which payload to deploy MME or DXD, adjusting its attack strategy entirely on its own inside the human body.
The drug stops being a static weapon. It becomes an active thinking agent. You don't just drop the software and hope it hits the target. You program the cure to adapt.